Good day, dear students. In current lecture, we will discuss unique family of bacteria, which is called Spiracetaceae, and all microorganisms belong to that family because of several reasons. And as it obvious from the name, all of them have spiral shape. Besides that, they have microaerophilic type of respiration. They have unique organelle for motility, which is called endoflagella. It is a type of flagella which is located within the cell and not visible during regular staining techniques. Due to complex structure of microbial cell, any spiral bacteria have long doubling time, and usually cultivation on culture media is difficult because microbes are poorly growing on culture medium, and we have biological method of cultivation, but again it takes a lot of time because of long multiplication of bacteria. And bacteriological method or microbiological method is usually not used for diagnosis of the diseases, and we will discuss further which methods are applied for identification of those bacteria. Besides that, bacteria usually have very thin cell wall, very thin microbial cell, and in regular staining techniques, it's poorly visible, and gram stain is rarely applied for that bacteria. We have specific methods to identify bacteria in microscope. Again, we will discuss in current lecture. The first genus we will describe and discuss is Trepanomaa. It has two main species, Trepanomaa keratinum and Trepanomaa pallidum, and the last one is subclassified into subspecies. Again, classification was revised recently for several times, and in some elder books you can find that Trepanomaa pertunia and Trepanomaa endemicum are described as a species, separate species of bacteria. And nowadays they are reclassified and they are suspected to be a subspecies of one Trepanomaa pallidum species, the main one. So bacteria is very long in size. It is forming spiral coils with a regular space between them, and bacteria is looking like a good correct spiral shape or corkscrew shape. And if you will apply gram stain, it is gram negative, but again, as it obvious from the name, pallidum originates from pale. It's very weak stain by gram stain, and it's easy to miss that bacteria in clinical sample when other cells are present in slide. And usually we have Giemse or Romanovsky stain or some other staining techniques which allows us to see bacteria much better. And in the center of the slide, you see that blue colored Trepanomaa. It is Giemse stain, that's why bacteria is blue. Later we'll discuss diagnosis, but one of important and unique factors in the structure of bacteria is a cardiolipin. It is a component of the cell which is used in identification of bacteria and diagnosis of the disease. It is a natural compound of antigen of Trepanomaa, and besides that, it is present in some other animal species and have very similar structure. So, as we said, bacteria have endoflagella, and the whole microbial cell is surrounded by mucous substance which resembles capsule, and it is very often called as an outer stitch and can be identified if required in that bacteria. As we said, bacteria is very difficult and poorly growing in culture medium. Trepanomaa was never cultivated in laboratory conditions in cell-free culture medium, and actually we have only animal inoculation as the only way to grow the bacteria. And again, as we said already, bacteria cannot be stained by regular techniques. To understand a little what is endoflagella, I will show you that picture in the right part of the slide. It is electron micro photography of the Trepanomaa. It has endoflagella, and with the arrow it is marked, you can see that one filament, muscle-like filament, is going through the whole cell and connecting coils between them. So due to that location, unique location of flagella, bacteria can rotate or flex different microbes have different number of endoflagella. In Trepanomaa, it is single one, and bacteria can flex or rotate by the whole cell. And in the lowest part of the picture, OM is outer membrane, IM is an inner membrane, and in the left picture, as you can see, it's actually not Trepanomaa, but all spiral bacteria have common structure. The same endoflagella is going through the whole cell, and we have the axial filament as the unique structure covering endoflagellas. And actually, bacteria have two membranes: cell membrane or inner membrane and outer membrane. And some space between those membranes is called as the periplasmic space. To see bacteria in not electron microscope, we have several techniques. Again, as we said, cultivation is difficult, and usually if you need to grow that bacteria by some reason, we have only rabbits as susceptible animals, and inoculation should be done in testicles, but method of course is expensive. Bacteria is slow growing, and a lot of time is required to obtain pure culture of bacteria. Again, not any laboratory can deal with rabbits. It is a specific laboratory who have them, and in routine practice that method is not used. Usually we will take clinical samples, some smear from the subject, and try to find bacteria here. But again, in clinical samples, a lot of other cells will be present, and we need to identify bacteria in detailed or precise manner. So we have three main techniques. One is a common stain used in regular microscope. It is silver impregnation method, when ions of silver are added to clinical sample, and they will not specifically bind to the cell wall of spiral bacteria, and bacteria will become thicker, and it will be easier to see that bacteria in microscope or even in regular light microscope. And silver, of course, it is a metal which not passing the light, and bacteria will be less transparent. So as you can see, that yellowish background is due to low concentration of silver. And in the places where silver accumulates in high amount, mainly it is microbial cell, you can see that different colors of brown, it is bacteria, and you can see the shape spiral bacteria, easy to identify, even so thin bacteria as a Trepanomaa. Two other methods require unique type of microscope. It is dark field microscope, which is usually available in routine laboratories. And in that case, you will have background very dark, even black, and bacteria, which is present here, will reflect the light in the microscope, and you will see that kind of glowing or shining of white color. It is a reflected light. And as you can see again in that sample in the center of the slide, we have another cells and some bubbles. Because it is a clinical sample, and something is present here besides bacteria. But anyway, bacteria is easy to see. And in the case of dark field microscopy, we will use liquid which is suitable to support the life of bacteria. And of course, you can identify motility of bacteria. It will move, rotate, or move through the slide, and it's easy to differentiate it with some maybe hair-like structures or some inclusions in slide dust particles, which can make diagnosis a little more difficult. And in the left slide, in the left picture of the slide, you can see immunofluorescence. It is a technique which requires UV or luminescent microscope. Bacteria is treated with antibodies labeled with fluorescent dye, and under UV light, they will glow in corresponding color, which dye is used to label antibodies, the same light you will see. So technique is known as DFATP method, direct fluorescent antibody test. And again, it's very specific. If in other cases you will see only the shape of bacteria, and you cannot be sure that it is really Trepanomaa. In the case of immunofluorescence, we always use antibodies against known bacteria, and if they bind it to microbial cell, it is for sure known bacteria, and you can perform diagnosis with the help of DFATP test without any other methods. Trepanomaa have a very wide range of diseases caused in humans, and usually they are classified into two main types: venereal and non-venereal. Venereal disease is called so because of venous gut of sexual relationships, and actually any sexually transmitted disease very often is called as a venereal disease. So sexually transmitted syphilis identified only in humans, and if you have one infected person, you always need to find another one, because other ways of transmission in the case of venereal syphilis are not possible. Only sexual contact, human to human. And it is one of the most important measures of prevention of the disease to stop transmission, because actually out of human body bacteria cannot survive for a long time. It's very sensitive to environmental factors. It will die very soon. So only direct contact can transmit bacteria from one person to another. In main number of cases, up to 90% of cases, entry gate for bacteria is genital organs or genital area. In rest cases, we have so untraditional ways as a rectum, anal area, anal region, oral cavity, and even nipples. So actually, any part which can be involved in sexual intercourse can act as an entry gate for bacteria. And actually, Trepanomaa, due to unique type of motility, do not require some damages on the mucous membranes. It can pass through intact mucous membrane, and usually it is observed in the site of the first contact. If the first contact was in some oral region, then in other areas, lesions will not be identified. And if you see initial lesion, it is always the site of the first contact. So any disease caused by Trepanomaa usually divided into three stages: primary, secondary, and tertiary. They have different appearance, but always have very similar pathogenesis, very similar reasons. And if you describe disease caused by Trepanomaa, you just can name phases, and it is important during your answer: primary, secondary, tertiary stage. In the case of venereal syphilis, primary syphilis. Every stage differs from other by unique type of lesions which are formed and can be identified in human body in different parts, but always it is unique presentation in clinical symptomatics. And in the case of venereal syphilis, that unique lesion is a heart chancre. It is small papule initially in the site of first contact. It will be red, elevated above level of the skin, maybe a little itching. With the time, it will break and form a kind of small ulcerated element which have lost sensitivity, have no blood vessels. It is shining, and base of that element of that ulcer is very hard. That's why it is called heart chancre. So chancre is a French word, and pronunciation is not English. It's not very correct to say cancre or chancre. It is heart chancre. So that element, that ulcer, will be covered with mucoid sticky discharge, and actually it is the site of intense and active multiplication of Trepanomaa. And that element is highly contagious. Due to local immune reaction, those people can have regional lymphadenopathy, and later with the bloodstream, bacteria can disseminate to other organs into the bloodstream. Usually that chancre is visible because, of course, people have them in some open parts of human body. But if it is some rectum or deeper area in the throat, people cannot notice that element is present because it's painless, it's not bleeding, it's not performing any discomfort, and it's actually very difficult to feel that something is wrong. Only if it is real enlargement and some discomfort is present. So of course, if those people will notice that ulcer appeared, they usually will go to the doctor and look for medical help. And if people are asocial, and usually venereal diseases are identified in a social population, some drug abusers or alcoholics, they can not pay sufficient attention to that ulcer. Some damage appeared, but they not care. So even without treatment, the chancre will heal in up to a month, and it's looking like spontaneous recovery because usually element heals without any lesions or some thin scar can be formed, but it's not bothering, it's not really visible, and people feel that they are fine, they are cured, and they will not go to the doctor if they even wanted but had no time or some other reasons. And the chancre will heal. And about 70 to 60% of population will have spontaneous recovery in the stage. So chancre will heal and bacteria will be eradicated. Immunity, if it is fine and not impaired, will eradicate bacteria, clear the bloodstream, and bacteria will not invade into organs, and spontaneous recovery is observed. About 1/3 of the patient will progress to secondary stage. So that is the common appearance of heart chancre. I will not show it on genital area by obvious reasons, but some unique location as a tongue or lips is fine to show here in that lecture. And in the right picture, you can see that it is deep alteration, and maybe skin around that ulcer will be edematous, red, elevated above the skin, and that skin can be little bothering the subject. But usually, the chancre is not bothering people unless it's visible and looking like awful. So, secondary syphilis usually appearing after some months after chancre heals. Some time required for bacteria to disseminate over the body and to multiply in significant amount. And the first usually 3 months after healing of heart chancre, no symptom is identified in those people, and that's why they're thinking that they are cured. But after some time, bacteria will go again to the bloodstream and spread over the body, and initially it will appear in the skin. You can see that the whole body will be covered with secondary lesions, and there are multiple. If chancre is usually one, and the only exception is HIV infection and some severe immunodeficiencies, where one subject can have several chancres. But in main number of people, it is always one. Secondary lesions are always multiple. They can cover the whole body. It is syphilitic roseola lesions. It's looking like a roseola or papillary element, can merge and form patches, and can be absorbed in mucous membranes as well. So every element appear in secondary stage is the site of multiplication of Trepanomaa, and any contact with that element is highly contagious. So subject in secondary stage is the most dangerous, and the stage can last even up to 5 years. And for the whole that period, people can infect other relatives, friends by contact with them. And of course, it is extremely important to recognize disease in this stage and to give proper treatment, to isolate the subject and give proper treatment. But again, if it is some extremely asocial person, and even that secondary stage will not make that person will to go to the doctor, even secondary stage will have spontaneous convalescence in, again, time period up to 5 years. And people again will have pseudo recovery because everything is gone, they're feeling fine, no symptoms are present, and people will think that they're cured. And actually, about 30% of people will be cured. Few elements of that unique roseola and papillary rashes. It can be identified in any part of the human body, and even in mucus. In the lowest part, in the lowest picture, it is a tongue covered with those patches, which have unique appearance like a blister of bubbles and elevated above the level of the mucous membrane. And we have that condylomata, unique lesion in area of genital organs. It's looking like overgrowth of the tissues, like papilloma or some cancer-like appearance. And as we said, about 30% of people will have spontaneous cure and even without the therapy, immunity will deal with that bacteria. So then we have some, uh, if it's so to say, problems in further classification, because different medical schools, different countries can identify and recognize different number of stages. So it can be 3, 4, even 5 stages of syphilis. So about 1/3 of people will progress to latent stage. It is called latent syphilis, when absolutely no symptoms are present. But if you will perform any serological test during some routine checkups, or person will be hospitalized by other reason, test will become positive. But if you perform an investigation, subject is absolutely fine. No damages, no rashes, no elements, nothing. And nothing is bothering the subject. So we call it latent syphilis, and actually in Russia, that stage is supposed to be a prolonged secondary syphilis. It is a part of secondary syphilis. But some countries say that it is a separate stage, it is third stage of the syphilis. So here you can have different names and classification of the stage according to medical school you will read article or book. So about 40% of ill people will progress to tertiary syphilis. It can be third or fourth stage, and that stage is characterized by appearance of unique lesions, which are called gumas. The TS type of granuloma resembles some mycobacterial one or other infectious granuloma, but gumas are lack of treponema. And actually, those lesions can be identified in any organ in any tissue, and slowly they're replacing organ tissue with empty area. Cavity will be formed, and tissue just destroyed. So usually, tertiary syphilis is classified into different types according to which system or organ is affected more than others. We have cardiosyphilis, we have neural syphilis, we have GIT syphilis, and some bone syphilis. But actually, it is just tertiary syphilis with one system organ affected more significantly than others. In that stage, people are usually not contagious because treponema is present in very few amount. It is trying to hide in the skin, in different tissues, but easily killed by macrophages. And that abnormal reaction, formation of granulomas, will be identified in people. So the last stage, it is late tertiary or quaternary syphilis, and in some even literature it can be identified as a fifth stage of the syphilis. In Russia we call it late tertiary syphilis because actually it is a consequence of tertiary syphilis, when significant amount of neural tissue will be involved and destroyed. People will have some complications and manifestations as a result of those neural tissue damage. And they can manifest with different paralytic disorders in extremities, in fingers, toes, legs, any area. And later, when central highest functions will be affected, they will have mental disorders. Usually, those people kind of old because late tertiary stage requires a lot of time to manifest and to cause that difficult disorders. And nowadays, the stage is extremely rare observed because of routine checkups and good methods of diagnosis and treatment. Syphilis usually treated before the stage. But in some even previous century, in 90s, we had some number of cases, even in our city in Smolensk, when those people were identified first with the syphilis in the tertiary stage. And of course, it is not possible to treat the disease already because it is mainly immune response, not infectious process, which is responsible for that unique damage of organs and tissues. One of the important problems in the case of venereal syphilis and actually any disease caused by treponema is extremely short immunity. Usually, in 1 or few days after convalescence, human became susceptible to the same bacteria already. And reinfection is kind of normal thing in those people. It is possible to have it even the next day after complete recovery. And it is the reason why, again, disease is circulating in human community and it's difficult to eradicate it completely. And it is extremely important to identify every contact person who was contacting that subject you have, because if you will treat your patient, that patient will go home and will have a contact with the same ill person and come to you in a few days or some in a week after that. You will discharge the subject. If you talk about diagnosis of the disease, we have two major groups of tests. They are classified into treponemal and non-treponemal. Non-treponemal tests are based on identification of that cardiolipin antigen. And as we said, it have common structure with some animal species, and usually we use cardiolipin obtained from beef heart or some other extractions from animals, because it have common structure with bacteria. But the cardiolipin not belong to treponema, and actually it will identify not directly treponema. The antigen can be observed even in human. And test is usually used as a screening test to remove negative people, because if test is negative, it is negative for sure. We have only false positive results. And second group of tests is treponemal tests. They're usually based on identification or use of antibodies against treponema. And those tests are more precise. They have much less number of false results. And usually we have the same fluorescent, immunofluorescent test or agglutination test, TPPA, to identify treponema in clinical samples. A few photos of that gumma, and the first one under that signature syphilitic gumma. It is initial appearance of gummatous lesion. It is overgrowth of the tissue, enlargement, local enlargement, and it is beginning of immune reaction. Later, it will be destroyed, necrotized, and concomitant flora, bacterial flora can add, and it can manifest with some purulent discharge, but later it will be removed completely, and you see that just some kind of ulceration, absence of tissue is observed. As we said, it can be located in any organ and tissue. Skin is just more indicative. And later those gummas will heal, or tissues around the gumma will heal. And you will have, like here in those pictures, just empty area instead of tissue. So usually nose affected, soft tissue of nose, or even bones of nose, and you see that holes in old men are present in nose instead of tissues. And if the tissue was formed in hard palate, you can see in the right part of the slide in the right picture, it is just a hole from oral cavity into nasal cavity, and it is open area because previously here we had the gumma which was replaced with empty area. Of course, disease is known from old ages, and we have a wax sculpture in museum which showing possible consequences. It's called One Night with Venus. Possible consequences, serious consequences of syphilis without proper treatment in ancient times. And now we have only some small empty zones, small holes, but previous years even half of the face could be destroyed by bacteria, and people lived with so ugly appearance. They have a lot of problems, of course, with common life, because no nose, no natural protection against bacteria, and a lot of concomitant infections can add, and people die because of some bacterial complications. We have unique type of congenital syphilis. It is a significant problem in social women when untreated women can transmit disease to their fetuses. And during formation of tissues, especially bones, fetus will be damaged, and those kids are delivered with significant abnormalities deformations of different parts of the body. In that picture, it is an Indian girl who have no nose from the very beginning of the life, and usually those kids are non-viable. They will die because of serious deformations of bone structures or because of concomitant infections. But if they will survive, they can live for some years. They will have that ugly deformation for the rest of the days. And of course, it is extremely important, and nowadays it is obligate measure checkup of or screening of pregnant women on syphilis. And the main difference of congenital syphilis, primary stage is always absent. Of course, fetus is infected through direct infusion of maternal blood, and that kid will have secondary stage of syphilis as the first one. And later disease will progress by the same principle, the same stages. And in severe cases, as you can see in that right picture, it is extremely ugly deformation when eyes are absent, teeth are absent, nose is absent, and actually that face is looking like someone took an eraser and removed some part of the face. So we have again some non-venereal syphilis, or even it's more correct to say that it is venereal syphilis with non-venereal ways of transmission. We have that syphilis economica. It is a unique type of disease when a bacteria is transmitted through non-direct contact in some public places, such as coffee, restaurants, and some other when maybe dishes are not washed properly or people sharing dishes. Or it can be transmitted even through wearing of common clothes when families are poor and some parents can give their pants or some other personal things to kids or to family members. And very rare cases it was described when even common transport such as buses or trams were acting as the place of that infection. When person, especially in secondary stage, is visiting public transport, they can touch some door hand or some other seats and other things, and next person who will have very quick contact, bacteria can survive for some seconds or even minutes, and that material can be transmitted to other people. So even through shaking hands, but again, usually when heart chancre is located in palm or in secondary stage when elements are single and poorly visible, people can shake the hand and disease can be transmitted from person to that non-sexual but direct contact. And actually, some liquids of human body can contain bacteria, especially if lesions are located here. And saliva or some mucus near the lips, near oral area, can contain bacteria, and disease can be transmitted through sharing some bottles with water or some food. One person bited and another one. So it's again kind of alimentary way of transmission, but again, it's type of direct person-to-person contact. And if women became infected after pregnancy, or maybe it is some donor breast milk, then disease can be transmitted through breast milk from mother or from woman to kid who was delivered healthy, but kid can become infected immediately after the birth delivery through eating breast milk infected with bacteria. Unique type of syphilis is professional syphilis. That one is extremely important to you because if you will remember that actually any lesion during primary and secondary syphilis is highly contagious, you will understand that it's obligate to use gloves and to use common measures of protection during any type of examination of the subject. Don't touch skin or don't touch some personal things of the subject. If you see the person have any element on the skin or mucous membranes, always use personal protection measures. And even some cases of infection during medical instruments or some material in laboratory practice were described when laboratory workers touched test tube, dirty one test tube, or touched slides containing infectious material with bare hands. And of course, bacteria is highly contagious. It can pass through intact epithelium. And doctors and laboratory workers can become infected if they're not following measures of common infectious protection. We have, and nowadays again, it is almost not possible, but still we have, historically, we have cases of transfusional syphilis when blood of donor was not checked correctly and bacteria was injected to healthy person from people with latent syphilis or from some unchecked donors in tertiary or other stages. And of course, nowadays, again, it is an obligate manner checkup of donors for syphilis, and that type of disease is almost not registered. If we'll talk about again preparations, immune preparations or biological preparations used for identification or prevention of the disease, we have several of them. It is already mentioned cardiolipin, which is identified in Trepanomaa as well as some animals. And actually, it is a natural compound of mitochondria, which is identified not only in beef heart, it is found in animal tissues and human tissues as well. And you see that in that picture, it is an ELISA test for cardiolipin antibodies, IgG, A, and M. But the main penalty of the test, it is fast, it is pretty sensitive, but it will show you presence of any type of cardiolipin. And false positive results can be observed even in some human diseases, such as diabetes mellitus, such as different type of cancers, autoimmune disease, when tissues of human body affected by incorrect reaction of immunity and internal containment of cells is released into environmental area, it can be absorbed in the blood. And mitochondria, even a human one, can give positive test with cardiolipin. That's why the test nowadays is not used. Previously, it was used as a screening test to identify negative people and then should not be studied further. But any person who is diagnosed as a positive with cardiolipin test should be re-evaluated with some additional tests. We have a separate preparation of treponemal antigen, which is observed obtained from treponemal cells. And it can be used for complement fixation test to identify presence of bacteria in human body or samples. And nowadays, still in some countries, Wassermann reaction is used, WR. It is a modification of complement fixation test. And again, the test is the same, non-specific, because it is still using cardiolipin antigen. And the test, if it is still used, it should be used only as a screening test. Again, if your laboratory, if your hospital will use Wassermann reaction is a test for identification of treponema, don't put diagnosis only by one positive test of Wassermann reaction. And previous century, previous decades, as you can see on that picture, the test was significant to put the diagnosis and to prescribe proper treatment. But nowadays, it is strict requirement that any cardiolipin test should not be used as confirmatory test. It is only for screening. So if the test is positive, or even in some countries the test is not used at all, you should perform some more detailed tests. And nowadays, all previously mentioned tests are replaced with that VDRL evaluation. VDRL is acronym from Venereal Disease Research Laboratories. In India, the test is used in main number of laboratories as well. It is the type of microprecipitation when reagent antibodies are used to identify treponema in human samples. And as you can see on the left picture, in the case of negative result, you will have absolutely clear area in the slide and the drop of liquid. Again, you need to mix two liquids, patient's sample and reagents from test set. And if you have a area which is absolutely transparent and clear, it is negative result. If you will have formation of clumps, some groups or kind of flakes, test is positive. In some test systems which are more sensitive, we can have weak positive, strong positive, or in main number of cases we have non-reactive and reactive result. So if cores or flakes are formed, test is positive. If liquid is transparent and colorless, or even if some color is present but it is homogeneous, you see that the whole solution have equal homogeneity and equal color. The test is negative. If some inclusions are formed, then you can say the test is positive. So as we said, vaccination is absent and prevention of the disease should be done by only non-specific measures of control. Use of barrier contraception, regular checkup of medical doctors, obligate checkup of people who are admitted to hospital or looking for any medical help, especially if it is some surgical intervention, checkup of pregnant women, and of course, if you identified one person, it is obligate to find all possible contact people, but of course it is extremely difficult because usually those people are social, they don't remember who they messed with, or they have multiple contacts and they will try to hide other partners from the main one. And sometimes they're not even looking for medical help. Of course, proper treatment of the disease should be given for any ill person, and nowadays we have very active antibiotics which can provide good efficacy with very short regimens, and they're available by the price.